Hello there, my name is Simone Reinwald. I'm a research officer at Alfred Health and Monash University in Melbourne, Australia. I'm here to talk about our recent letter to Allergy with the title Expansion of Phenotypically Modified Type 2 Memory B Cells After Allergen Immunotherapy. Fairly recently, an IgG-positive memory B-cell subset was defined, which is expressing high surface levels of CD23 and IL-4 receptor alpha. These cells were termed type 2 memory B cells, and they are shown to be expanded in allergic subjects. Now, since we also previously observed an increase in allergen-specific memory B cells that do express high levels of CD23, IL-4 receptor alpha, and CD29 after successful allergen immunotherapy, we now wanted to investigate allergen-specific type 2 memory B cells in two allergic cohorts. One cohort is the ryegrass pollen allergic cohort, and one is the bee venom allergic cohort. Both cohorts receive immunotherapy for the duration of 4 months. As you can see in those little images on the left-hand side, the ryegrass allergic cohort receives sublingual immunotherapy, and one arm only receives pharmacotherapy alone. Now, we collect peripheral blood mononuclear cells before and after 4 months of allergen immunotherapy to identify our memory B cells via flow cytometry. For the detection of our allergen-specific memory B cells, we use in-house produced allergens. For the ryegrass cohort, that is the LOLP1, the Lolelium perenna 1 protein, and for the bee venom cohort, that is the Apis mellifera 1, APM1 protein. Those proteins are coupled to fluorescent strep evidence, which is then creating a fluorescent tetramer. We use those fluorescent tetramers to distinguish our allergen-specific memory B cell, as you can see in this flow plot here on the right-hand side. Within the allergen-specific memory B cells, we identify then the CD23 positive population. Now, at the bottom left side, you can see in that figure that within the allergen-specific memory B cells, a significantly higher proportion of cells express CD23 than within the total memory B cells, and this was seen in both the ryegrass pollen and the bee venom allergic cohort. So this shows an association of the type 2 phenotype with allergen specificity. We then examined the impact of allergen immunotherapy on type 2 memory B cells. Looking at allergen-specific IgG4 levels in serum in the left figure, we can see that for the ryegrass pollen cohort, LOLP1 specific IgG4 levels increased after 4 months of immunotherapy, but not in the group that only received pharmacotherapy alone. A similar trend in red is seen in the bee venom cohort, where we also see a significant increase of serum IgG4 after 4 months of immunotherapy. In the right figure, we are now looking at the absolute counts of allergen-specific type 2 memory B cells in both cohorts. And we can see a significant increase in allergen-specific type 2 memory B cells after 4 months of immunotherapy in both the ryegrass and the bee venom allergic cohort. This trend was not seen in the pharmacotherapy-treated ryegrass patient alone. So, as sublingual immunotherapy for ryegrass pollen allergy induced an upregulation of CD29 and IgG4 on our LOLP1 specific memory B cells, as we've shown in a previous publication, we now wanted to evaluate here whether the expanded allergen-specific type 2 memory B cell populations were phenotypically altered. In the left-hand side, we can see a significantly higher percentage of allergen-specific type 2 memory B cells expressing CD29 after immunotherapy, both for the ryegrass pollen cohort and the bee venom allergic cohort. We do not see this in the pharmacotherapy-treated ryegrass patients alone. On the right-hand side, we're now looking at the IgG4 frequency, within allergen-specific type 2 memory B cell. And we see that this is increased following 4 months allergen immunotherapy for both the ryegrass pollen cohort and the bee venom cohort. And again, not for the ryegrass pollen cohort that receives pharmacotherapy alone. In conclusion, we show for the first time that the increase of allergen-specific IgG4 over the course of allergen immunotherapy is directly associated with the expansion of type 2 memory B cells that express IgG4. We show an upregulation of CD29 on type 2 memory B cells after 4 months of immunotherapy. Our results look at a time frame of 4 months though. Immunotherapy regimens usually take 2 to 5 years, so it is now of course of great interest to see how this initial expansion of type 2 memory B cells plays out over a longer duration of time. So to determine this, long-term studies are needed. I conclude here and I say thank you for your attention.